Okay, good afternoon everyone, good afternoon. Okay, so friends, let's finish the little left part of this particular cell communication. By the cell communication chapter will be completely done. Okay, now let's start. The communicating, see, I hope you all people remember, friends. So first we have studied, good afternoon, Saima, Sumaya, Akshay. Okay, we have studied first the four type of the CAM molecules. After studying the CAM molecules, friends, we started studying about the junctions. Is it? We studied tight junctions, we studied adherence junction, we studied desmosomes, we studied focal adhesions, we studied hemidesmosomes. So after that, the next one is, friends, what? Communicating junctions. Once we finish the communicating junctions, we will move to the last part, that is the extracellular matrix proteins, which are extremely important and which we have to read and remember compulsory. Okay, now let's start. Communicating junction. Friends, the name itself very clearly indicating, right? Communicating, which means it is helpful in the transport or movement of ions from one cell to the another cell. Good afternoon, Annie. Continue. So that is what we can call communicating junctions. Good afternoon, Anjana. So the examples of the communicating junctions are what, friends? Gap junctions as well as the chemical synapse. So let's talk about both very elaboratively. If you very quickly recall, we just started discussing about the gap junctions and we ended the last class. And friends, please kindly do not get confused with this point. Where the gap junctions are present majorly, friends, listen to me carefully. Gap junctions are present between the two cells, right? And I want you to look into the diagram very carefully, friends. This is the membrane of the cell 1 and this is the membrane of the cell 2. You people can observe it very clearly. You know, listen my statements. When we observe, we feel that the two membranes are coming very close to each other. We can observe that the two membranes are coming very close to each other. But friends, in reality, there is a small gap is present. Friends, listen to me very carefully. Between the two membranes, friends, there is a very small gap is present. How much gap is present, friends? Approximately 3 nanometer gap is present. So I want you people to remember that 3 nanometer gap only can be covered by this particular gap junctions. And every one of you listen to me very carefully, friends. What does this particular gap junction is made up of? Everyone listen my words very carefully, friends. The gap junction exclusively made up of a protein called as connexons. Connexon. How does the connexon is formed? Connexon, you can imagine that connexon is a kind of polymer. Okay, it's not completely polymer. Connexon is a major protein which is forming the gap junction. How does a connexon will be formed? Connexon will be formed by the 6 connexins molecule. Kindly do not get confused with this. 6 connexin molecules come close together and they are going to form one connexon molecule. Kindly don't get confused. Look into the diagram very carefully, friends. Look into the diagram, everyone. This is called as connexon. Correct, na? Friends, this connexon is made up of 1, 2, 3, 4. So 1, 2, 3, 4, 5, 6. 6 connexins will come close together and they can form one connexon molecule. Do not get confused with that. And friends, everyone has to remember that this particular connexon, listen my words very carefully. These connexins are of same type also. And friends, these connexins are of different type also. Friends, if the connexins are of same type, we will be calling it as the homomeric connexins. Friends, if this particular connexins are of different types, we can call it as the heteromeric. One point is very clear, whether it is the same type of the connexins or the different type of connexins doesn't make any sense. Basically, all the connexins come close together and they will be involved in the formation of this particular connexon. Continue, Shafia. Good afternoon. And listen to me very carefully. With this particular connexons, do you think every molecule or every ion can be transported? No, that is not possible. Friends, every ion or every molecule cannot be transported. Only a particular ion or a particular molecule can be transported. Kindly do not get confused with that. You can see a 100 kilodalton, 1000 up to 1000 kilodalton also it can easily transport. It can easily transport through this particular connexon. If it is more than 1000, they cannot transport at all. So what is the point every student has to remember? The transport specificity, listen my statements very carefully everyone. The transport specificity is determined by the size of the ion or molecule that is getting transported through the connexon molecule. Kindly do not get confused with this. Listen to me friends, the gap junctions were first discovered where? The gap junctions were first discovered in the myocardium because of their properties of electrical transmission between the adjacent cells. Kindly do not get confused with this particular point. And here comes the most unique statement you people has to remember. By using a gap junction, what kind of ion or what kind of molecule can be transported? That is the point I want you people to remember. Listen to me very carefully friends. By using the gap junction, keep it in your mind, I told you long molecules or the large molecular weight molecules cannot transport. I never say that lipids can easily transported through this particular gap junction and proteins can be transported. No, that's not possible. Only I can use the word. What is the word I can use? I can use the word that they can majorly involved in the transport of monomers. What are monomers? Monomers are single units and they are not having large molecular weight also. So they are having the small molecular weight and the size is also small. As a result friends, they can easily transport through this particular gap junctions. Everyone listen, cells share their small molecules such as inorganic ions, sugars, amino acids, nucleotides, vitamins. Not only this, and I want you people to remember, even intracellular mediators such as cyclic AMP, inositol triphosphate, you know right, the powerful secondary messenger cyclic AMP and IP3, even friends, they are also can be easily transported through the gap junctions, but not what is not getting transported, that is more important. What is not getting transported is the macromolecule. Kindly do not get confused with this particular point. And friends, I have clearly told to all of you that this is called as one connexon. And what did I tell you? One connexon is made up of how many connexins? I said to you, one connexon made up of 6 connexin molecules. And I want you people to remember, every connexin molecule can transpan the plasma membrane 4 times. Kindly do not get confused with this particular point. That is what exactly friends, we will be calling it as the tetraspan. Once again listen to me very carefully friends. One is the connexon. One connexon made up of 6 connexins. One connexin can transpan the plasma membrane 4 times. Kindly do not get confused with this particular point. Do not worry about this. I want you to compulsory remember what is connexon and how many connexins that it is made up of. Okay. So the best example for the gap junction what we have studied is the connexon. And another best example of the gap junction, what you can take friends, plasmodesmata. Friends, the moment you heard the name of a plasmodesmata, two statements has to come in your mind. What are the two statements that will come in your mind? That is, one is called as symplastic pathway. First point you have to keep it in your mind. And friends, plasmodesmata, second point you have to remember that it is acting as the cytoplasmic bridge. Kindly do not get confused with this particular point. What friends? One is it will show the symplastic pathway, and most importantly friends, it is acting as the cytoplasmic bridge. What do you mean by the word cytoplasmic bridge? Which is involved in the transport of ions or molecules from one cell to the another cell with the help of this particular bridge. And I want you people to remember, while studying the gap junction, I told you na, every ion, every molecule cannot transport. I have told you very clearly one important statement, that is, listen to me, transport specificity is determined by the size of the ion or size of the molecule to be transported. And I want you people also remember that this particular plasmodesmata also never allow every ion or every molecule to be transported. That's the point I want you to remember. What I want you to remember is that this particular plasmodesmata, friends, will allow only a particular ion or particular molecule having a fixed size or a fixed shape, and most importantly fixed molecular weight also. Kindly do not get confused. All the students, please kindly look into the diagram. This is how exactly the plasmodesmata look like. Look into this. Here exactly the plasmodesmata is present. So it is connecting two cells, and it is involved in the transport of ion or molecule between the cell. And friends, what does this particular plasmodesmata is composed of? What does this particular plasmodesmata is made up of? Everyone keep it in your mind. The plasmodesmata is made up of desmotubule. Right, and the plasmodesmata is made up of smooth endoplasmic reticulum. So listen my statements very carefully. Desmotubule, I can use the word, it's a kind of a monomeric protein. Many desmotubule protein come close together, they will be make up of this particular plasmodesmata. Desmotubule as well as the pieces of the smooth endoplasmic reticulum also make up of this particular plasmodesmata. Kindly do not get confused with this particular point. Okay, now listen. In the communicating junctions, friends, listen, we have taken two examples. One is the gap junction, we have taken an example, and another very important one we are going to take for the example is the chemical synapse. Chemical synapse is the most important one for the exam. I want everyone to remember. Okay, let me explain you elaboratively. Just listen to me for 10 minutes. Just listen to me for 10 minutes. Two principal kinds of synapses are there. What are those? Everyone listen to me very carefully without confusion. One we can call the electrical synapse. Another one we can call the chemical synapse. Friends, I want all of you has to understand the points very clearly. Whether it is the, you listen my statements very carefully for 10 minutes without confusion. Whether it is the electrical synapse or the chemical synapse, doesn't matter. Both synapses are involved in what? Both are involved in the movement of the ion or movement of the molecule. That is the major function. Yes or no? Of course, yes. Now, all of you listen to me. Then what is the difference? The moment you heard the name of a chemical synapse, what is the statement has to come in your mind? Here the movement of the ion or the movement of the molecule can be exclusively mediated by the neurotransmitters. Can be exclusively mediated by the neurotransmitters. Kindly do not get confused. And here Here also there is a movement of ion or molecule, there is a no doubt about it, but it is basically mediated by the electrical current, or there is a voltage difference will allow the movement of ion or molecule. All the students look into the diagrams very carefully, friends. This is presynaptic membrane, correct na? And friends, this is what? This is the postsynaptic membrane. Friends, what is the major difference between the presynaptic membrane as well as the postsynaptic membrane? Everyone pay utmost attention and listen my statements very carefully. When I use a word presynaptic membrane, see, this is one neuron and this is the second neuron. So the ionic movement is occurring between one neuron to the another neuron via, all the students look into the picture very carefully, friends. What are all these? These are all the gap junction channels. They are nothing but what channels? They are gap junction channels. So friends, based on the voltage difference, listen my statements. Based on the voltage difference, the gap junction channels will be closed open, will be closed open, will be closed open. All the time you remember that particular point without fail. Now friends, the ions are getting flow through the gap junction channels. Kindly do not get confused with this particular point under any circumstances. Here the ionic movement is based on the voltage difference. Now the most important one which we have to remember is the same ionic movement with the help of a powerful neurotransmitter like acetylcholine, GABA, 5-HT, adrenaline, noradrenaline, serotonin. There are many neurotransmitters are there which are involved in the movement of the ion for many purposes. Say smooth muscle relaxation, for the skeletal muscle movement. Friends, listen my statements. For many kind of responses, we require the movement of ions. An example, the muscle has to be relaxed, or a skeletal. See here, I am moving my muscle. If I am moving my muscle, there should be a compulsory requirement of flow of ions. So that flow of ions which are required for the movement of the skeletal muscle, which can be mediated exclusively by this particular neurotransmitter. Just listen. All the students listen. I'll show the diagram in the next slide also. This is the presynaptic membrane. This is the postsynaptic membrane. All the students look into the diagram very carefully. Friends, you are all able to see, right? This is, what do you call it, friends? This is, you will be calling it as the synaptic vesicle. What you will be calling it as? You will be calling it as, friends, synaptic vesicle. All of you please listen to me very carefully. This synaptic vesicle, everyone pay attention. This synaptic vesicle contains what? They contain abundant amount of the neurotransmitters. Kindly do not get confused with that particular point. Now this synaptic vesicle which contain abundant amount of the neurotransmitters, everyone listen. Whenever they come and fuse the membrane, see here, they fuse the membrane. The moment they fuse the membrane, you can see here the green color one. Green color one are the neurotransmitter. Friends, I want you to listen to me very carefully. All the neurotransmitters will come and associated where? Friends, look into the diagram. The neurotransmitters will come and associated with the receptors which are present on the postsynaptic membrane. Once again listen, they will come and associated with what, friends? They will come and associated with the postsynaptic membrane. Listen my statements. The movement, friends, understand, the moment neurotransmitters are associated with the receptor, immediately what will happen? Understand, friends, immediately there will be a opening of the ion channels. Immediately there is a opening of the ion channels. The moment there is a opening of ion channels, there is a movement of the ion will be occurred. Now what is the statement every student has to listen? Here every single neurotransmitter can act as a ligand. Once again understand my statement. Every single neurotransmitter can act as a ligand. For what purpose? Every neurotransmitter can act as a ligand for the ion channel. Once the ion channels get opened, then only ionic movement will occur. Let me explain you the same thing little more elaboratively. Look into the picture. Friends, all of you understand. Imagine this is the presynaptic membrane, this is the postsynaptic membrane. Understand what exactly I am saying. The point is here, the movement, the ionic movement is occurring from the presynaptic membrane to the postsynaptic membrane. We all people know it very well. So friends, here comes an important statement is that if the ionic movement wants to occur, first of all, it is compulsory, it is necessary, it is mandatory that two presynaptic membrane and postsynaptic membrane has to be associated with each other. I mean to say both has to be joined with each other. Now the point is very clear how they will join. The question is how presynaptic and postsynaptic membranes can be associated with each other. Friends, the point is very clear. They basically associated with each other with the help of different type of CAM molecules. Kindly do not get confused with this particular point. Listen to me carefully. Chemical synapse is the junction between a nerve fiber and a muscle fiber, or between the two nerve fibers, through which signals can be transmitted by the release of chemical transmitter. Okay, listen to me very carefully. What happened? Why the people are going back? Okay, no problem. Listen. No worries. Look into this. This is the presynaptic membrane. This is the postsynaptic membrane. Everyone pay utmost attention and try to understand the concept very clearly. You are all able to see here, friends. There are abundant amount of the synaptic vesicles are present. What is that particular synaptic vesicles are consist of? They consist of, friends, abundant amount of the neurotransmitters. We all people know it very well. Correct na? Abundant amount of the neurotransmitters are present. Now you people listen to me carefully. First of all, if the ionic movement wants to occur between the two neurons, there should be a contact should be established between the pre and post. How? Please keep it in your mind. With the help of cadherin molecule and with the help of Ig superfamily molecule. These two are major. What is that? Cadherin. I hope you all people remember also. Cadherin can mediate what kind of interaction? Homophilic interaction. We read it very clearly. And immunoglobulin superfamily CAM molecule also mediate what kind of interaction? Undoubtedly we know that they will also follow the homophilic interactions only. We all know that very well. But cadherin and IGSF is there, but there should be another interaction which is also very important, I can say. The presynaptic membrane and postsynaptic membrane associated with each other by neurexin and neuroligin. Kindly do not get confused with this particular point. Listen my statements very carefully, friends. One is what? Neurexin. Another one, what do we call it as, friends? Neuroligin. All the students look into this. Here you can see now, friends, neurexin can act as a ligand. Neurexin can act as a ligand. And friends, what about the neuroligin? Friends, neuroligin basically can act as the receptor molecule. Kindly do not get confused with this particular point under any circumstances. Neurexin will be the ligand and neuroligin will be the receptor. Do not worry about, do not forget this. You people look into this, where the neurexin is present on the presynaptic membrane, where the neuroligin is present on the postsynaptic membrane. So there should be three interactions compulsory: neurexin, neuroligin, cadherin and IGSF superfamily CAM molecule. With the help of these three interactions, presynaptic membrane and postsynaptic membrane will be associated with each other for the longer period of time. Kindly do not forget it. Okay, now whenever the synaptic vesicle come and fuse the membrane, what happen friends? I want all of you has to look into the picture very clearly friends. These are all the ion channels. You can see here, to the ion channels who will come and attaches? Neurotransmitters will come and attaches. The moment neurotransmitters will come and attaches, ion channels will be opened. Then there will be a movement of ions will occur. In this picture also you can see it very clearly. The same thing. All of you look into the diagram. This is the synaptic vesicle. What does the synaptic vesicle contain? Neurotransmitter. The neurotransmitter come and fuse to which membrane? Which membrane it will come and fuse? The presynaptic membrane. The moment it fuses with the presynaptic membrane, there is a release of abundant amount of the neurotransmitter. Neurotransmitter in this picture you can see little more elaboratively. They will go and associate. See here, they go and associated with the ion channels to open or close them. So once the ion channels get opened friends, there will be a movement of the ions will occur. It's a very simple point. By that ions can be transported. Okay friends, there are many type of ion channels are there. One we can call the gated ion channels, another one we can call the voltage gated ion channels. See, I want you people to remember one simple point. When I use a word gated ion channels, everyone listen my statements very carefully. They are gate like structure. Listen friends, they are gated like structures. Friends, these gated like structures will be open, will be closed. Everyone listen. The gated like structures will be opened or will be closed with respect to the, everyone listen, with respect to the attachment of a ligand. So the ligand may be anything. The ligand may be growth factor. Most of the time, what is the ligand? The ligand will be neurotransmitter only. Do not get confused friends. It's a very important. Listen to me carefully. The gated ion channels, they may be open or they may be closed. Depending on what? Depending on whether the associated receptor has been activated by the binding of its specific ligand or by change in the transmembrane electrical potential. Most of the time they will be opened and closed with respect to the attachment of the specific ligand only. Let me give you one wonderful example here. All of you pay attention. Example of a ligand gated receptor channel is the, everyone listen, nicotinic acetylcholine receptor. Kindly do not get confused with this. What do we call it as? Nicotinic acetylcholine receptor. Every one of you listen my statement. The receptor channel opens in response to what? Neurotransmitter acetylcholine. So when the acetylcholine come and attaches to that receptor, then it will open, then only it will allow the movement of ion or molecule. All the students look into the diagram very carefully friends. You can see right now the gate is closed. Why the gate is closed? You can look into the picture friends. All the students listen, look into this. This is the area where the ligand will come and attaches. This is called ligand binding site. And what will be the ligand here? The ligand here specifically acetylcholine molecule. Now since there is a no ligand binding, that's why friends there will be a no opening of the channels, then there will be a no movement of the ions also. Now you only listen friends. Whenever there is a association of the acetylcholine molecule, everyone pay attention. Whenever acetylcholine molecule will come and attaches, what will happen? What will happen immediately? Whenever there is a acetylcholine molecule will come and associate, look into this. Whatever the channel so far, whatever the gates so far closed, it get open. Now you can ask me a very valid question. Sir, how they are getting opened? What is the cause here? The cause is very simple. There will be a conformational changes. The point is very simple friends. There will be a conformational change. Okay, all the students listen to me very carefully, friends. There will be a conformational change occur. Because of this particular conformational change, what is basically happening, friends? The ion channels will get open. Now you see here, abundant amount of ions are coming inside, sodium, calcium, and whatever has to come on. And we all people also know very well, the ion flow should not be continuous, because friends, we know very well that every single ion has to maintain homeostasis. What do you mean by that? Every single ion has to maintain the ionic concentration in the cell. So once the abundant amount of the ions enter inside, we have to close it, right? Immediately what happened, once again the conformational change occur. Whatever the ion channel that is opened so far, it will get desensitized. Desensitized means upon structural change it will get desensitized. Now the ligand will be dissociated, that is acetylcholine will be dissociated, and they will come back to the closing position. This is how, friends, the ligands will be helpful in the movement of the ion or molecule. Very, very simple point. So by this we have finished the second part, that is the junctions also. I told you now, the cell communication subunit, we have to read 3 subparts. One is about the CAM molecules, another one is about the junctions, and the third and the most important we have to study is the ECM protein. These 3 we have to study. Is it? Of course, yes. Now all the students listen my statements very, very carefully. Now we are going to read the last part, ECM. So friends, I already told you how we can expect the questions. You know, we can expect the questions in the 4 marker only. I am talking majorly. Only from the CAM molecule we can expect. ORR, only from the junctions we can expect the 4 marker. ORR, only from the extracellular matrix we can get the 4 marker. If not, most of the time, you know how they will frame the question. They will take 1 or 2 example from CAM, 1 or 2 example from junctions, 1 or 2 example from ECM, and they'll make 1 4 marker question, either in the form of a statement or in the form of a matching. You don't get any kind of experimental, analytical, graphical, only 2 ways, either statement or matching. That's all, nothing else. Okay. So that's why this is a completely theory concept. I want you people to read it compulsory. Okay. Now there is a extracellular matrix. What are the ECM? Now let's start. See friends, the moment you heard the name of a extracellular matrix, what is the first function you people has to remember? I'll tell you one common point, friends. Every one of you listen my statements. Extracellular matrix, friends, remember the common line. It can act like a organizing center. Everyone listen the statement. It is acting as a organizing center. In the developmental biology we have studied, if I want to form a complex organism, first it has to start with what? Cell. Correct now, friends? Listen everyone. Cell has to be organized into tissue. Tissue has to be organized into organ. Organ has to be organized into organ system. Now finally organ system is giving rise to a typical organism. We all know that. So how does this organization is starting? This organization is basically starting from this particular matrix only. Kindly do not get confused with this particular point. In animals, the extracellular matrix helps organize the cells into tissues and coordinates their cellular functions by activating intracellular signal pathways. If you can ask me, so what is the fundamental functions of the extracellular matrix inside the cell? Extracellular matrix proteins are required for cell growth, proliferation, as well as many gene expression. This particular extracellular matrix is required. Kindly do not get confused with this. Okay, now the point is clear. Extracellular matrix is a packaging material or a organizing center. Point is here, what does this extracellular matrix is made up of? That is the question. Friends, extracellular matrix is not made up of one or two proteins. They will be made up of, friends, many proteins. But among all the proteins, I can tell you, friends, these four are the most important ECM protein. Now you can ask me, sir, why you are calling only these four are the important, why not other? The reason is because they are ubiquitous in nature, which means they are present everywhere in the ECM protein, and they plays a key role in most structurally also, and they will provide mechanical strength also. Now you can ask me, see, ECM or basal lamina, anything is same, don't get confused here. I want you to understand most of the extracellular matrix components are located in the basal lamina. So ECM and basal lamina is same. Don't get confused. It will be present on every cell, friends, every cell. It's around the muscle cell, it's around the fat cells, and it's around the epithelial tissues, it surround the blood vessels, what not. In everywhere, in every cell, the basal lamina is present. Muscle cell, fat cell, and epithelial tissue. Kindly do not get confused with this particular point. Now listen to me very carefully. I have given you one clear cut point. The moment you heard the name of extracellular matrix or basal lamina, what you have to remember is that it is the one which will provide the mechanical support. Do not get confused with this particular point. Okay. Now, I told you it required for cell growth, cell division, cell differentiation, cell migration. For everything, the basal lamina is required. Now, this basal lamina made up of multiple proteins. Among the multiple proteins, four proteins are major proteins, which we are going to discuss very briefly and very elaboratively. Most of the questions will be appearing from here only. Type 4 collagen, laminins, intactin. Most of the time in the exam also they won't use the word intactin, they will use the word nidogen. So you people always remember as the nidogen only. Fourth one is the perlacan, and friends, fifth one is the glycoproteins. Kindly do not get confused with this particular point. Don't forget it. Okay, all of you pay attention, friends. Type 4 collagen, it is a trimeric molecule. Everyone listen. I will explain one by one, just keep it in your mind. Among all the type of proteins present in the basal lamina, the most important one is the type 4. Now you can ask me, sir, why you are calling only type 4? Because there are many different type of collagens are there. There is no doubt about it. Because collagen present on the tendon, ligament, smooth muscles like uterus, it surround the blood vessels. Many places collagen is present. Then why we are specifically talking about the type 4 collagen? Because friends, type 4 collagens are abundantly found in the basal lamina. Most of the ECM, it consists of type 4 collagen only. Now you can ask, you can understand, or you can guess one point. Does all the ECM proteins, will they arranged in a order? Will they arranged in a order? Never ever. They never arranged in a order. You know how they arrange, friends? They arranged in a group. And I can use the word that there is no particular position to associate with each other. All the students look into this, friends. By looking the diagram, you people can come to one conclusion that, listen my statement, one ECM protein providing the binding site for the another ECM protein. Everyone listen. So my left hand is one ECM protein. My right hand is one ECM protein. My left hand, which is a ECM protein, which is providing the binding site to the another ECM protein, which is my right hand. See here. So what you have to understand, all the ECM proteins are associated with each other. Kindly do not get confused with that. Understand the statement very correctly. Now you can see, friends, This is what? Type 4 collagen. Correct na? Of course, yes. This is the type 4 collagen. And this is what, friends? Laminin. And this is what, friends? Intactin. The pink color one we can call perlactin. What you people can observe, you can't tell after which protein which is associated. See, all are associated in a group because one ECM protein is providing the binding site to the another ECM protein. Okay, friends, let's start discussing one by one elaboratively. Okay, let's start with the first one, collagen. Everyone listen. Collagen, keep it in your mind. Collagen is a family of fibrous glycoprotein that are present in the extracellular matrix. Now friends, please remember the statement without fail. Collagen is a primarily produced by which cells? Collagen primarily produced by fibroblast. What is fibroblast, friends? Which is a typical connective tissue. Friends, please don't forget, okay? Collagen is majorly formed by the fibroblast, which is a major connective tissue where it is present, that are present in the extracellular matrix. Major one, it is present in the extracellular matrix, synthesized by the smooth muscle cells also, as well as the epithelial cells also. How many type of collagens are there, friends? All the students, please listen my statements. More than 20 type of collagen molecules are there that are participated in the formation of the extracellular matrix. Kindly do not get confused with this. And every student pay attention. What are the common features of the collagen? Everyone listen to me very carefully, friends. Do not get confused. The moment you heard the name of a collagen, what is the common point you have to remember? Collagen is a trimeric protein. What protein? Collagen? It's a trimeric protein. And I want you people to remember without confusing. Collagen is a type of a protein which is globular in nature also, at the same time which is in the helical in nature also. Now how is that possible, you can ask me. Now all the students listen to me very carefully, friends. See, imagine this is one chain, this is the secondary, and imagine, friends, this is the tertiary. Correct na? Collagen is what, friends? Collagen is a protein. So protein having what domain, friends? Protein having N-terminal domain. Correct na, friends? Protein having N-terminal domain, and friends, protein having C-terminal domain also. You all people know it very well. Protein having N-terminal domain, protein having the C-terminal domain. What is the statement I want you to remember? N-terminal and C-terminal domain of the collagen is the globular in nature. Then what about the remaining portion? Friends, all the students remember, remaining portion will be helical. So that's why, you know, if you observe the collagen at the terminal positions, at the N-terminal and C-terminal, they are globule, very smooth. But if you see the middle, it's a very tough, very, very tough. It's a, fibrillar, rigid cable-like structure. How strong the cable is? In the same way, the collagen is also very stronger, because you can imagine 80% of the collagen is fibrillar in nature only. Only the N-terminal and C-terminal only globular in nature. Don't get confused with this particular point. Now listen. All of you understand type 1, type 2, type 3. I want you to remember entire information, friends. Type 1, type 2, type 3 are fibrillar collagen as assembled into rigid cable-like fibrils. That's why we can't separate the collagen very easily, and we can't break the collagen is also very, very easily. Now you listen, friends. This is the point you have to remember, where the type 1, type 2, type 3, type 4, where they are actually present. Those things I want you people to remember. Type 1, they are abundantly present in the tendons, ligaments, and bones majorly. And friends, please keep it in your mind, type 2. You know, right? Most of the people will eat the meat. When they are eating the chicken, they will crush the bone and they'll eat the cartilage or the bone marrow present inside. Is it? So why? Because it is highly proteinaceous in nature, which is extremely good for the nails as well as the hairs. Most importantly for the tendon, ligament and joints. Most importantly. Why? Because friends, they contain type 2 collagen only. Don't forget. It represents more than 50% of the protein in cartilage. It is also used to build the notochord of vertebrate embryos. Do not forget that. And friends, when you look into the type 3, what is the point I want you to remember? Type 3 strengthen the walls of hollow structures, like very important, arteries, intestine and uterus. You all people know that artery is a smooth muscle, intestine is very smooth, uterus is more smooth muscle. This is, listen my statements carefully. Even though they are very smooth muscle, they are stronger. They are highly strengthened muscle. Why? Of course friends, because of the presence of type 3 collagen only. And the most important collagen, that is the type 4 collagen, which is present in the extracellular matrix also, which is present in the blood capillaries also. I want you to remember friends. And even friends, which is present in the glomeruli of kidney also. Kindly do not get confused with this particular point at all. Please do not forget it. Okay. Now listen my statements very carefully. All the students look into the diagram friends. You can see here, N-terminal domain is the globular, C-terminal domain is the globular. And friends, this is the collagen, the type 4 collagen. Remaining, you can see now friends, remaining entire region is a very tough, very tough fibrillar collagen. Some are fibrillar, some are non-fibrillar. But you remember, most of the middle part will be the fibrillar region only. Only the extra N-terminal and C-terminal domains are the globular. Don't forget that friends. So first type of the ECM protein is the collagen. Now the second type of the ECM protein that we have to discuss is the proteoglycan. Friends, you people can understand by name itself, right? Proteoglycan, which means it is made up of protein plus sugar. Correct na? I want every one of you has to remember what are the name of the sugars that will be added to the proteins. Most of the time friends, polysaccharides only will add. Most of the time polysaccharides only will be adding. So what do we call it, that particular polysaccharide chains, or what do we call that sugar specifically, which is adding to the proteins? Everyone keep it in your mind. We will be calling it as the GAGs. Glycosaminoglycans. Shortly, what do we call it as friends? GAGs. What are the, most of the time, what kind of GAGs or what kind of sugars that will be added most of the times? I want you people to remember most of the names without fail. One is called as uronic acid. Another one is the hyaluronic acid. I want you to remember friends, very important, these are all. And there will be a addition of D-galactose, and there will be a addition of N-acetyl glucosamine. Then there will be a addition of N-acetyl galactosamine. These are all the different type of sugars that will be adding to these kind of proteins. Sorry, these are all the various type of sugars that are adding to the proteins which are present on the ECM protein. Now the point is very clear. Now you can ask me, sir, how all the sugars will be associated to the proteins? Undoubtedly friends, there is no doubt about it. Covalent linkages only. There is no doubt about it. Of course, there should be a amino acid which is involved in the linkage. All the students please kindly look into the diagram carefully. This is the core protein. The blue color, whichever I am marking with the red one, this one. This is the core protein. Friends, you all people can see here, these are all the different type of sugars. Now, how all these different type of sugars will be associated here with the help of a amino acid serine. Now, you can ask me, sir, why only serine amino acid? Because friends, in the active site of the protein, everyone listen to me, in the active site of the protein, what do you mean by active site of the protein? The area or the point at which sugar will come and attaches. That is what exactly, my friends, we will be calling it as the active site. Kindly do not get confused with that. There, this is the serine. You can understand the point very clearly. This is the serine. To the serine, different kind of sugars will be added. So friends, what is the point you have to remember? What kind of glycosylation it is? That is the O-linked glycosylation. You have to remember. What kind of statement or direct part B question we can expect in the exam? Which of the following glycosylation can be mediated by the glycoproteins present in the ECM? So all the proteins which are present in the extracellular matrix, whenever they are associating with the sugars, they will always follow O-linked glycosylation only. Kindly do not get confused with this particular point under any circumstances. I have told you friends, different kind of sugars can be added. Galactose, N-acetyl-glucosamine, N-acetyl-galactosamine, glucuronic acid, iduronic acid, xylose, D-glucose. Many sugars will be added to this particular core protein. Now friends, among those, some of the major sugars, hyaluronone, heparin sulfate, chondroitin or dermatine sulfate, then followed by the keratin sulfate. Is there any necessity to remember the structure? No, there is no need to remember any structure at all. What you have to remember compulsory for the exam is what that particular sugars are actually made up of. That is the only point I want you to remember. What actually that sugars are made up of. That is what I want you people to know very well. Hyaluronone. See friends, here nothing to explain. I want you to remember compulsory linkages. Compulsory. Hyaluronone made up of D-glucuronic acid and N-acetyl D-glucosamine. These two are the sugars which are connected with each other by beta-1,3 glycosidic linkage. You have to remember. Now, how does the heparin sulfate? You have to remember here, nothing to explain. Glucuronic acid or iduronic acid associated with the N-acetyl N-sulfoglucosamine. Why we are using the word N-sulfo? There is a addition of the sulfur group also. Keep it in your mind in the heparin and chondroitin and keratin. Heparin, chondroitin and heparin, there is a presence of the sulfo group. Don't forget this, and the linkage will be beta-1,4, either alpha or beta-1,4, both. Chondroitin sulfate made up of iduronic acid and N-acetyl D-galactosamine. The linkage is alpha-beta-1,3 linkages. Keratin sulfate made up of D-galactose and N-acetyl D-glucosamine. The linkage is beta-1,4. Here nothing to explain. I want you people to remember only name of the sugar, sorry, name of the protein, what kind of sugars that they are formed and what is their linkage. Compulsory you have to remember. Okay. Is it clear friends, everyone, till this particular slide? Clear? Okay, perfect. Okay. So friends, listen everyone, in the developmental biology chapter, I told you na, ABC model in the Arabidopsis thaliana and antirrhinum we have to read, and we also have to read the limb development. Only 2 subtopics are there. It will take 3 to 4 hours. And in the biochemistry, I know that we are left with 5 topics. What I do is biochemistry left topics and developmental biology, that 2 topics, just take 4 hours. I will do at a time. Okay, I will do at a time. Okay, from tomorrow onwards I will do the genetics. Okay, the moment genetics get over, I will do the left part of biochemistry and that two topics of the developmental biology. Okay, so from tomorrow only I will start the genetics. Then at last I will do the cell biology and evolution topics. And most important one, the unit 9 diversity of life forms. I will explain you the most important topics which were frequently asking in the exam. Okay, okay friends, that's all for now. I'll meet you in the tomorrow class. Okay. Okay friends, that's all for now. Okay, we will finish the cell communication in the next class and we will start the host pathogen interactions. By that the unit 4 will be completed. Very simple. Okay. Okay friends, I'll meet you in the next class. Okay, thank you all. Thank you. Please revise it without fail. Without fail, please revise it. Okay, please revise. Please revise.